Nonmetric multidimensional scaling, or NMDS, is one of the most flexible ways of examining unconstrained patterns of how species are distributed across sites. Until recently, this form of ordination wasn't done much because it took more computational firepower than was generally available. Now, however, that is no longer generally the case, and so it has become one of the most commonly used forms of ordination. NMDS is an unconstrained form of ordination, like PCA that we did earlier in the semester. That means that in an unconstrained ordination, you're looking at variability within a single data set, like a site-by-species data set. Unlike other forms of ordination, however, uniquely in NMDS, you specify how many axes or dimensions you want to display that variability across. And this can be tested to determine whether or not the number of axes that you chose does a good job of representing the variability in species across sites. You start with creating a distance matrix or a dissimilarity matrix using any distance metric of your choosing. And then the ranks of the dissimilarities among the objects are calculated. The original distances of pairs of samples in your original distance matrix are ranked, and this becomes the target rank order. In other words, what you're aiming for. You're going to compare the configuration of the ordination to this target. So for the number of axes that you specify, and that can be any number from 1, which is ridiculous. You would have all of the variability in a complex data set on one axis. Don't do that. Up, 2, 3, 4, and once you start getting above 4, then things start getting very unwieldy, difficult to interpret, and you're in danger of overfitting. So somewhere between 2 and 4 axes. For that number of axes that you specify, the sampling units are assigned ordination scores. So they're mapped onto those axes of variability. Then a Euclidean distance matrix is built based upon where these objects are now arranged on these axes. The NMDS algorithm then finds a configuration of objects in that chosen n-dimensional ordination space that best matches the differences in the ranks between the original distance matrix and then how it has been ordinated. Then what's called a stress parameter is calculated. Stress is a measure of the lack of fit between object distances in the ordination space and the calculated dissimilarities from your distance matrix. And the stress indicates how much the ordination scores have to be adjusted to achieve monotonicity. And by monotonicity, I mean as an axis increases, the response variable increases, or as it decreases, it decreases. So stress of this object, of the ordination, tends to increase with your sample size and with the number of response variables because you have a more complicated set of variables, more complicated data set. And just like in life, high stress is a bad thing. So this process is iterated until an ordination with minimal stress is found. So to put it another way, NMDS is an iterative search for the best position of n entities on k dimensions or axes that minimizes the stress of the k-dimensional configuration. In terms of assumptions and limitations of NMDS, there are relatively few. It doesn't require that your data have a normal distribution. It doesn't assume that there are linear responses between your variables. And it doesn't mind outliers, although it's best to cull your outliers first. Outliers can drive an ordination in a certain direction. And you can use any distance or dissimilarity matrix. And so that makes it very flexible, and it's one reason why NMDS is so widely used. However, there are some important limitations that we need to talk about. The first of these is that NMDS is not an analytical process that will get you to a single unique solution. Instead, it's an iterative algorithm where the initial estimates of positions along of the samples along your ordination axes are adjusted to minimize the stress until further iterations don't see a significant improvement. And so NMDS is sensitive to the initial positions of the samples along the ordination axes, and that is a random assignment at first. And so NMDS can sometimes settle into a local optimum that is not globally the best solution. It's just the best solution that's better than nearby solutions. And so because the initial sample ordination scores are randomly assigned, every time you run an NMDS with a different random number seed to start the process, you will get a different solution. And so because it doesn't settle on the ordination, this is seen as NMDS's chief weakness. But remember that NMDS, or any ordination, the point of that is to elucidate patterns in your data. And so if it produces a solution that elucidates patterns, then it has been successful. You rerun it, you get a different ordination. It should presumably, if those patterns are true, it presumably should allow you to discern those same kinds of patterns in your data. However, you need to be advised that when you run it, you get an answer and then you do it again and you're like, hey, I didn't get the same answer. That's inherent to NMDS. In terms of other considerations, it is computationally demanding, particularly if you have very large data sets, although this isn't the problem that it was just a few years ago. Nowadays, most desktop computers can handle NMDS on pretty large data sets. And in terms of minor points, the solution can be influenced by very abundant species. So what do you do about that? Well, there's not any sort of perfect solution. What you might do is try running it with and without including very abundant species, because just like outliers, the presence of very abundant species can drive an ordination in a certain pattern, a certain way. NMDS does not work well on small data sets, meaning fewer than 10 samples, because it creates randomizations that are too conservative and that produce configurations for which NMDS will tell you that the stress is 0, which is not realistic. So what you should do is interpret your results very cautiously if you have a small data set. And similarly, if you have data with lots of zeros in them, NMDS doesn't produce, it doesn't handle it very well. And what you should try to do is do randomizations, do the iteration for a very large number of iterations to try to see if there is some consensus that emerges. But once again, be sure to interpret your results very cautiously if you have a data set with a lot of zeros in it. Okay, so now let's go through an example of NMDS. And in the online lesson, I talk about 3 different commands and packages that you can use to do NMDS. One is MetaMDS in vegan, there's IsoMDS in mass, and there's NMDS in lab DSP. And we'll have examples of all of these. For any of these, the first step is for you to calculate a dissimilarity or distance matrix. And so what you have to do is then make a decision, what distance metric should I use? So in the online notes, the example we worked with was the Bryce Canyon vegetation data set. So when you look at those data, what you should do is go through this logical process to decide what distance metric is going to be the most appropriate. We did this in the ordination introduction lecture as well. So to go through this logical process. For example, in the case of the Bryce Canyon data, first off, it's a homogeneous data set, meaning that it is comprised of a single data type, like numerical data. And so that eliminates our need for the Gower metric. The data aren't counts in this case. Remember, it's percent cover. And so that eliminates Steinhaus. There are lots of zeros in the data set, which means that Euclidean, Manhattan, and Canberra are not going to be your best choices. And it also should cue you in that perhaps NMDS might not be super great at handling these data, since there are lots of zeros in it. Mahalanobis wouldn't be appropriate for this, and chi-square really de-emphasizes abundant species, of which there are several in this data set, and so maybe that's not our best choice either. But again, if you really want to downplay the role of very abundant species, then you might want to use a chi-square metric. But for any of these this logical thought process, that's what you're doing. You're going through and saying, this one's not great, this one's not great. And so for the case of the Bryce Canyon vegetation data, I chose to use the Bray-Curtis metric. That is the most commonly used distance metric for NMDS. It's the default method in vegan, in fact. So this is what we did: create the distance matrix, and then you perform the NMDS on that using, in this case, it was the LabDSV program package that we used first. And then you calculate the stress. And so the stress value, in my case, remember that every time you do NMDS, you'll get something different. In my case, the stress value was 19.27838. That's what I have in the online notes. So now the question is that high stress or is that low stress? We want it to be low. So what is a good value? What is low? In order to determine whether a value of stress is high or low, we have to compare that value to stress values from other ordination configurations of these data using different numbers of axes. So the default number of axes is 2. And so that 19.27 value that I got was for a K of 2. And so what you do is you have to play with the number of axes to see how the stress values change. And that's the main unique feature about NMDS. Unlike other ordination techniques, it allows you to perform the ordination with various numbers of axes and pick the best. But this should be done with care. Choosing too few dimensions, too low of a value of K, will force multiple axes of variation to be expressed on a single ordination dimension. But if you choose too many dimensions, that can cause a single source of variation to be broken in a way and expressed on more than one dimension. The higher the value of K that you ask NMDS to work with, the lower the stress is going to be. And you can really start to get into problems if you just look at stress and say, I want to minimize my stress value. So what we're going to do in the next few slides is go through a process where we are trying to minimize stress, but weigh that against the number of axes that we need to do that. So an appropriate number of dimensions should be determined by examining stress versus number of dimensions. And sometimes this is most easily done by producing a plot because you can visualize it. So one type of plot that you can produce is called a scree plot, where you plot stress on the y-axis and the number of dimensions on the x-axis. And what you're doing is you're looking to see where the resulting pattern shows an elbow. In other words, the greatest drop or change in stress from one value to the next. So in the case of this scree plot, the first two axes produce a greater reduction in stress than other dimensions. And so in this case, we can conclude that a two-dimensional solution would be best for this data set. But most functions in R don't produce a scree plot. They produce a different kind of visualization that's called a stress plot. And it shows scatter around the regression line between interpoint distances in the ordination against their original dissimilarities in the distance matrix. And what you're doing is you're looking for tight scatter around the regression line. In this example, the regression line is in red and the points are in blue, and so you're looking for tight relationship between the points and the line. So that indicates that the reduced number of dimensions from the ordination does a good job of representing the original dissimilarities of the data. In this example, this example looks okay in this stress plot. So determine what the best value of K is to minimize stress. To do that, you need to examine multiple dimensions to find the optimal number of reduced dimensions. The default value when you're doing an NMDS in these packages is K of 2. So in my example that I worked with in the online notes with the Bryce Canyon dataset, when I boosted K up to 4, the stress dropped from 19 point something to 10.433. This is to be expected. Stress naturally decreases with an increasing number of dimensions. But keeping the number of dimensions less than or equal to 4 is advised to minimize the possibility of overfitting. So, at this point, to settle on a suitable number of axes based on low stress, now we've got, okay, I'm going to pick, a K of in this case, K of 4 gave me a stress value of 10.43. I need to know whether or not that 10.43 is good or not. It's the lowest I could get, but is it good enough? And that's what we'll cover in the next slide. In the online notes, I have 3 authorities with rules of thumb about what should be considered from excellent to poor stress values. And you can see here that even though the value that I got of 10.43 was low and was parsimonious, it was low relatively speaking, it would be seen as being fair to poor by Kruskal and flawed by Clark, and definitely not good to excellent by Zur et al. So that may seem disheartening, but most good, meaning tidy and interpretable, ecological community data sets have solutions of stress between 10 and 20. So these rules of thumb tables that I have in the online notes are really based on statistics and not on biology. So the fact that I got a value that is 10.43 is one that I would say, okay, I can work with that. That is acceptable. So we'll keep the K of 4 because that's the most parsimonious number of dimensions that minimizes stress. I could increase K to a number larger than 4, and that would drop my stress, but I would be in danger of overfitting the data. So keep it parsimonious. Where do you get the single greatest drop? The next number of dimensions. So we'll keep K of 4. Now we need to plot the ordination. In NMDS, the resulting ordination axes have no particular order of importance. Unlike PCA, for example, where the first axis represented the greatest variance in the data, then PCA2 was the next most, and PCA3 was the next most, and so on and so forth down the line. In NMDS, they simply represent the best possible mapping of positions. They do not indicate the percentage of variance explained by each axis, again, unlike PCA. And the axes do not have a direct interpretable relationship to the response variables. They simply represent a best fit of patterns of redundancy in the original data set. So because the ordination axes in NMDS are built to reflect patterns in the original data but are not calculated directly from the data because NMDS works on ranks, the axes themselves have no intrinsic meaning and there's no order to them. So axis 1 is not necessarily more important than axis 2. The default plot is to graph the first two axes, as is done here. Most ordination methods are analytical and result in a single unique solution to a set of data. In contrast, NMDS iteratively seeks a solution and stops when an acceptable solution has been found or after some pre-qualified number of attempts. As a result, an NMDS ordination is not a unique solution, and a subsequent NMDS of the same data will likely result in a somewhat different ordination. Therefore, I find NMDS plots to be rather uninformative. To evaluate whether an NMDS is extracting stronger axes than expected by chance, then a randomization test is done. We start by examining the correlation between the calculated dissimilarities and the plotted values. And after all, that's what it's trying to maximize. And we can do that with LabDSV's ordcomp function, and we generate a stress plot like this. And so for the four-dimensional solution that I had in the online notes, this is the resulting plot. Notice the correlation coefficient here. It's 0.946, which is very strong. The cloud of points isn't quite linear, but remember, an ordination is not a cartographic projection. The correlation coefficient R is for linear relationships, and so you really ought to examine the correlation for the first two default axes. And so when we do that, that's this, and in this case the value turns out to be 0.875, which is still quite high, indicating a good fit. So remember that our stress value for the original ordination with the default of K of 2 was 19.278, which according to Kruskal, Clark, and Zur et al. Was a poor value, but it was still one that was evidently better than random. Okay, so now that we have a reasonable ordination of the variability in our site by species data, we can take the axes from this unconstrained ordination, and then use them as dependent variables to examine the influence of environmental variables on this variability, just like we did in using the results of a PCA in a constrained ordination. With PCA we did RDA. And so we can do something similar to this in NMDS itself. For example, to examine the effect of soil depth from the site by environment dataset BryceSite.R, the influence of soil depth on the distribution of plant species by sites, then this is what you can get. This is an example that I used in the Introduction to Ordination lesson. And in the video for that lesson, I explained that NMDS likely wasn't an appropriate choice for this specific analysis because in examining whether soil depth could explain variation in the site by species data, because depth is a categorical variable and thus isn't on a continuous gradient. The ordination shows the overlap in ellipses here indicates that either soil depth is not a factor that accounts for differences in plants by site, or that NMDS isn't an appropriate choice for this type of analysis. So this is an important point. Just because you can get an analysis to work and can produce a graph with it, that doesn't mean that it is an appropriate analysis for your data or for your questions. Now let's do a demo of MetaMDS from package vegan on the same Bryce Canyon data with the same Bray-Curtis distance metric. One nice thing about this function relative to NMDS from LabDSV is that MetaMDS automatically transforms your data if needed. However, I have found that MetaMDS is much more prone to not finding stable solutions, stable stress solutions, and giving you a message of no convergence. So what you can do is you can examine various numbers of values of K and look to see where do you get the biggest drop in stress, and that allows you to determine, okay, what's the value of K that I need for my ordination. For the value K equals 2, the default K equals 2, the best stress I got, in my case, again, was 14.05. MetaMDS, by the way, reports stress as a proportion, so you have to multiply the result by 100 to get a value comparable to the rules of thumb we covered earlier and comparable to output from other functions. So recall that earlier we got, with NMDS function from LabDSV, we got a stress value of 19 point something for our two-dimensional solution, or at least I did. Remember that NMDS uses random configurations to start, so your results may differ. So one nice thing about MetaMDS from vegan is that you can use a function to determine the optimal value of K. And so in this case, what I did was I examined values of K from 2 through 5. And these are the resulting values here. This is the stress for K of 2, K of 3, K of 4, and K of 5 down here. And what you're looking for is where do you get the largest relative drop-off. So in this case, the difference in stress from K equals 2 to K equals 3 was nearly 4 points. Versus if we go from K of 3 to K of 4, we only reduce it by about 2, and as we go from K of 4 to K of 5, we reduce it by about 1. And so what this indicates is that a K of 3 here is the most parsimonious solution. All of these values of stress are acceptable, but the biggest drop-off was from K of 2 to K of 3, and so K of 3 is going to be the best solution. Recall that before, when we did the same data set, when we used NMDS from LabDSV, we determined that K equals 4 was the best number of dimensions to use to minimize stress. So this illustrates, again, one of the peculiarities of NMDS. There is no unique solution. So in this case, for the MetaMDS, with a K of 3, our stress value was 10.35%. So now let's make a stress plot. Another nice thing about MetaMDS is that its stress plot comes with both nonlinear and linear R-squared values, and I think that this is slightly more intuitive than just R, little r. Little r is the correlation between the predicted values and the observed values of y, whereas R square is the square of this coefficient and indicates the percentage of variation explained by your regression out of the total variation. MetaMDS also includes the regression line in red, and so it's much easier to see, visualize. Okay, so in this case, I would be happy with this outcome. So to summarize, the basic principles behind NMDS are: select an appropriate number of dimensions or axes, seek low stress, and then you can use a randomization process to compare your real data to a null model and avoid unstable solutions. So now let's take a look at some examples of how you can do this with IsoMDS from package mass. To illustrate IsoMDS from package mass, I decided to use a different data set. two different data sets actually, the hseer.csv and hseer_ph.csv files. hseer has abundances of plants collected on 10 transects named T1 through T10. And these transects represent a gradient of pH values, and those data are in hseer pH. If you look at those data sets, however, you'll see that the sites, instead of being rows, are as columns. And so we need to transpose these data in order to make them in a form that R is happy with. Okay, so you go through a logic process to decide what kind of dissimilarity or distance metric is going to be appropriate for these data. And homogeneous data set, eliminate scour, lots of zeros, so on and so forth. You go through this process and you wind up with some choices of Bray-Curtis or Hellinger or Steinhaus. And since Bray-Curtis is usually the default method because it's so flexible and appropriate for so many data sets, we're going to use that. So Bray-Curtis metric was used for this, and eventually when you go through the process and you find an optimal number of solutions and so forth, you do the IsoMDS on this, and when you plot it, you get this, which is, to put it gently, uninformative, even though it had a very low stress value. There is separation of sites, but only on one axis, and the low stress value is probably because only one axis was being involved here. In situations like this, the distance matrix that you chose is usually the culprit. And so even though we went through a logical selection process, this ordination isn't informative. And so what do? Well, let's default to the second most commonly used distance metric, which is Euclidean. And this is what you get. The result is informative even though its stress value was slightly higher. So this example illustrates how your choice of distance metric, even when done logically, can still result in an uninformative solution with NMDS. But remember that the goal of ordination is to reveal patterns. And when you continue with this particular example and conduct a correlation analysis between the values of axis 1 and pH, you'll see that it is strong and significant. So in this case, even though Euclidean wasn't my first choice, it produced something that looks logical, and statistically there was shown to be a significant association with the presumed environmental variable driving differences in plants across these transects here. So now let's take a look from the empirical literature at some studies that used NMDS. On April 20th, 2010, the Deepwater Horizon oil rig exploded in the Gulf of Mexico, killing 11 people and spilling over 4 million barrels of oil. That's over 168 million gallons, making it the largest marine oil spill in history and one of the largest environmental disasters in American history. Despite a massive cleanup effort, about 22% of that oil remains in the Gulf with presumed environmental consequences. In this study by Mason et al., they examined microbial communities in marine sediments at 64 locations along an oiling gradient. They examined microbial diversity in terms of OTUs, operational taxonomic units, as opposed to species. And they also looked at gene activity, particularly for genes that are known to be involved in the breakdown of hydrocarbons. So in this complicated multi-paneled figure here, panel A shows their NMDS plot, indicating associations between environmental variables and ordination axes. Now they didn't specify which distance metric they used. They simply stated that they used vegan. And so I assumed that they used the Bray-Curtis metric because that is the default in vegan. The three most abundant species, OTUs, in the contaminated samples are represented in with arrows here. And samples that exceeded the EPA's aquatic benchmarks for polycyclic aromatic hydrocarbons are denoted with these red circles out here. And you can see that the stress value is quite good. Again, in certain packages, they represent stress as a decimal, and you have to multiply it by 100 to get something that is like the rules of thumb expressed by Kruskal and Clark and Zur et al. Okay. In panel B, concentrations of various contaminants are indicated by bubble size and contour lines. In C, panel C, the rarefied abundance, meaning they underwent rarefaction, the rarefied abundance of two microbes, or operational taxonomic units, are shown with, again, with bubble size and contour lines indicating areas of abundance. From this NMDS, they concluded that there were significant differences in microbial communities and in gene expression in sites with presumed different levels of lingering contaminants in the Gulf of Mexico. The next two slides are from a study by Ushio et al. They collected insects from the field and found that each harbored millions of microbial cells on the body surface. A laboratory experiment showed that the microbial community composition on a flower's surface changed after contact with an insect, suggesting that microbes are transferred from the insect to the flower. So in this figure, it shows NMDS ordinations, which they conducted with Bray-Curtis similarity and showing 95% confidence ovals of the microbial communities from three different insect pollinators and from flower surfaces. In panel A, they have Xylocopa, which are carpenter bees. In B, there's Bombus or bumblebees, and panel C is Apis or the honeybees, and their associated laboratory control flowers. And each one of these panels is the same. It's just that each of the target species is highlighted in each one. And from this, you can see how each pollinator harbors a different microbial community. When those microbes are transferred to a flower, it can indicate which bee species are visiting that flower. Furthermore, they determined that those microbial communities differed from those in other environmental samples, such as in lake water or soil, as shown in panel A. In addition, the microbial community composition on insect bodies showed a degree of species specificity, as shown in panel B. So, they used NMDS plots to examine the microbial communities recovered from insects, flowers, and other environments, which allowed for some inferences to be made about species interactions. So for your assignment, you're going to be working with the grassland community data and the site by environment data associated with it from the plot.metadata file. And you're going to use MetaMDS from vegan. That is probably the most commonly used function and package to do NMDS in R. To answer the following questions. Remember that MetaMDS automatically does any transformations or standardizations if needed, and so you're going to determine what did they do, what was needed. Then look at the stress value, and is that value good? Justify that. Determine the optimal number of dimensions based on stress, and then redo the NMDS by specifying that value of K. What's the stress of this new NMDS model? And it should be lower than what you had before. And then assess your ordination's goodness of fit with a stress plot and interpret it.